features, you can have multiple proteomic features. And looking across all of that starts to help untangle some of that heterogeneity. So what we're seeking to do here is to capture variations that are introduced by genes and their mutation. So do you know the difference between germline and somatic mutations? So cancer, you've got an extremely heterogeneous system because of various somatic mutations that are coming into that tumour because of errors in the chaotic environment, essentially. So again, understanding the mutational state of a cell can have an influence on how that cell behaves. I'm going to show an element in a little bit where we've looked at, it's not single cell data, but we've looked at the effect of p53 mutations on a quantification of the p53 pathway in breast cancer. So we can also get variation introduced by microRNAs, non-coding RNAs, long non-coding RNAs. Now these things are not necessarily coding proteins, but they can have influence on what is on transcription. So microRNAs modulate genes by blocking and stimulating the transcription of the gene. You also get methylation. So genes are suppressed or stimulated based upon the level of methylation. So methylation is silencing genes. So you can have expression of a gene, but unless you look at its methylation, that gene could have a strong signal, but it might be methylated. So we have to consider that level of the epigenetics. We also have copy number variation, so genes can have multiple copies. That sends more signal. We can have translation, so errors can creep in translation. Proteins may be created. We get protein expression. Then on top of that, we start to get PTMs, so post-translational modifications. So if we can encapsulate those, then we start to understand how the protein signal is being modulated and changed. And then last of all, we get metabolites. And the metabolome is important, and that's kind of the end product. In fact, I would say the protein to the end product, the metabolome is a byproduct that's kind of tagged onto that because it's the proteins that are doing the functional work. The metabolites are a result of the protein's action. My view is that metabolites are much less diverse than proteins, and that occurring as a result of biological processes in the protein. We can also look now at spatial omics. So, have any of you heard of nanostring DSP? So nanostring DSP, you have a slice of tissue, you create regions of interest, and you get counts of sequence. I think last year it may have changed, it become even more. You've got measure essentially 18,000 genes on a region of interest. They're moving towards it being a deeper sequencing, even though where you measure the whole transcript. So you've got potentially transcript level information as well as gene level information for a region of interest. So nanostring have just been taken over and acquired by Bruker. So what did Bruker make? Sorry? In the context of this course, what did Bruker make? Mass spectrometers. So Bruker make mass spectrometers. Bruker are interested in spatial transcriptomics. So you're starting to get a potential towards spatial proteomics as well there, because you have a bead-related approach which measures transcripts, gene products, at the mRNA level. But you could quite as easily have a bead product which measures proteins. So then you have spatial proteomics using a bead-based approach, which is like a protein array, and has the capabilities of a protein array to measure lots and lots of proteins with great sensitivity. So spatial is important. The next layer after spatial is temporal. So if you've got time series data, these things are all varying in time. Yep. As I said at the beginning of this, consider systems biology or biological systems to be a concert where all sorts of things are playing together. Some things are influencing other things are coming in temporally at the moment and leaving, becoming louder and quieter. And it's an interplay between all of these things in the state. And multiomics, as its higher goal, is seeking to try and measure all of that. So we've got single cell omics and we've got temporal variations. And then, of course, all of this is happening through a population of individuals. We're not just modelling one person. Every individual has their own unique history. They have a lifetime of accumulated mutations in some cases. They have a particular diet. They have a particular ethnicity. One of the statements made about proteomics is you could, by looking at blood or serum proteomics, you could probably work out what somebody had for breakfast, because it's such a dynamic state. And that is a positive in that it's really useful data, but it's a negative in that it's introducing lots of variations into the profiling. So what really I'm interested in, and I suppose it's where the subject is kind of heading, is systems biology. And systems biology is the mapping of things over space, over time, at an interaction level. To truly have systems biology, we need that multiomics data over space and time. So we could ask the question, what is the interplay between multiomic elements? How are they interacting across cellular populations, across time, and differentially between a disease state and a non-disease state? So in an individual, we could look at tissue from a healthy part of an organ versus a diseased part of an organ, assuming there's no field changes. So a field change is where the disease is causing a change to the molecular profile of the tissue surrounding it. So we have to be careful of field changes in this sort of study. But through untangling that, we can start to understand what drives disease, what makes it tick, what's the basis, what's the cause of disease. So what happens then at the network pathway level when there's interplay between molecular features? So, as I said, by looking at a map of the pathway, by modelling that, you can get a quantification of the pathway. You can quantify that pathway across multiomics levels, but how do things change? If there's a particular mutation, how does that impact upon that pathway? If it's a different time of day, how does it impact upon that pathway? If you have different population groups, how does it impact upon that pathway? What's the effect of indels, insertion deletions in the genome, on that pathway? All of these things potentially have to be considered or could be considered. Because if we understand the system and how the system changes, we understand the various colours, for want of a better word, of disease, and how that spectrum of disease changes. So to illustrate this, there's a piece of work which I've done, I'm going to show you, which is modelling the p53 pathway in breast cancer by look and quantifying that model, that interaction model, in different mutational states. So what impact does the mutational state have on the pathway? So before, we perhaps might have just looked at a pathway not considered a mutation. But in reality, if you've got a pathway that the core element of that pathway is mutated, it's not going to behave in the same way. So by looking at wild type P53, by looking at loss of function P53 and missense, also nonsense P53 and missense P53, we can see how does that impact across the pathway. How does it impact on its stability and what features are coming out. So there were components of the multiomics data. I'll just zip through this. We've got the genome, we've got the transcriptome. Where are the limitations? Well, we've mapped the genome, but we haven't mapped it across the whole of human diversity. Now there are programs developing, every country has its 100,000 genomes, but historically genomes have been mapped for Caucasian people in the UK, US. India now has a 100,000 genomes program, which is developing. The Chinese have got a 100,000 genome project. More diversity is being mapped because there are different splice variants, different insertions, deletions that occur in different populations. We did some work on TB in China. A few years ago, we developed a signal based upon a South African and Caucasian population, and it turned out that one of our genes for a Han Chinese population had an insertion. So when we were doing PCR, we ended up getting 2 gene products for that gene insertion, which means that we were getting 2 signals instead of 1. So we had to resequence that gene for a Han Chinese population so that we could get a contiguous product. Contiguous measurement. So the genetics is important. The transcriptome, big problem with the transcriptome is ontology. Now we need to do more and more functional gene studies. So many genes, if you look at their ontology, they're biased towards cancer, or even more so breast cancer and lung cancer. That's because that's where they have been studied the most, but the functionality isn't truly represented by the ontology. There are gaps in the ontology. There are functions which need to be fully defined. So this is one of the problems with a lot of databases, is the database is incomplete. We don't have a full ontological representation of everything. So be careful of ontologies, because one case in point, if we look at Panther ontologies, in most gene lists that we look at, we see Alzheimer's disease and we see Parkinson's disease represented, even if it's cancer, which is a concern. But the reason for that is that they've basically lumped in so much functionality into Alzheimer's and Parkinson's because they don't really fully know how it's working. They're hedging their bets. So it's over-biased, the ontology, towards that disease state. It's not a real representation or unbiased representation. So ontologies are useful, but they are incomplete. The other thing is, with RNA sequencing, are all of you familiar with RNA sequencing workflow? Do you know the bioinformatic workflow to go from FASTQ files through to TPM values or RPKM values? Yeah. Well, when you look at the transcriptome, you can not only get gene level information, You can also get transcript level information. So that will give you the different transcript forms of the gene and the amount of signal coming from those. So some genes will have 15 transcripts in them. And across a population, you can have multiple transcriptional forms, which represent splice variants of that gene, essentially. And if you want to go even further, you can get exon level information for that gene as well, which gives you even more information. So we don't have to just look at the gene level, we can look at the transcript level. And in fact, I've got a project which I'm running at the minute where I'm looking at the relative contribution of multiple of the different transcriptional states to a gene and predicting that to understand what impact that those transcriptional states are having on a pathway. Because there's maybe different behaviors depending on the splice variant of the gene. So the spliceome is another level of complexity, and we don't really fully understand what that's doing. So that's another area of exploration. There's always more work to do. We can always go deeper, and this is the nature. In 1922, a group of physicists said, "We know everything there is to know. Science is dead." Then they discovered radiation. And perhaps it was before the 1920s, but there was a group of, I think they were either American or British physicists who were quite boisterously said and arrogantly said, "We know everything there is to know." But the world is such a complicated place, there is so much more to know, and we can go deeper and deeper. And that's, you are young, I'm less young, this is your future. There are an awful lot of things you could look at in the future, and there will be more things to look at as well. So, never think we know everything. The other thing, which is a gap in our knowledge, is there is a disconnect in multiomics between the message coming from the gene, mRNA, and protein expression. So if you look at multiomic studies and look at a set of gene messages, looking at mRNA from RNA-seq, and then look at protein expression, there is a block of proteins and genes that turn up. About 60-70% both will turn up and be going in the same direction. There's another 20% where there's no connection whatsoever. So we find the gene product, from the mRNA, but we don't find the protein doing the same thing. And then there's a smaller slice, even more, where the gene product goes up, i.e. there's more transcriptional message, but protein expression goes down. And we don't know why that is. But it suggests that these things are not directly linked, but there are other things like methylation or influences of other genes and other proteins on that system that are changing that regulatory direction. So we've still got to disentangle that. The problem is if we use fold change as a measure of importance, we will not disentangle that, because it's about correlation, it's not about magnitude of expression. So that's another challenge which we are yet to meet. The proteome is challenging. There are 14 orders of magnitude of expression of the protein in the human body, going from probably albumin on one level to proteins that are expressing at sub-picomolar levels. At the minute, we can probably measure 7, maybe 8 orders of magnitude with proteomics. With array-based methods, protein arrays, that might go up to 9. With some of the new mass spectrometry methods, that goes up to 9 as well. But we are missing things. We don't measure the whole proteome, so we have gaps there. That's something which will change over the future. Instrumentation will get better. So through building libraries, we can more accurately find these things, and if we know what we're looking for, we can more accurately measure them. So the coverage of the proteome will increase. As I said before, there are few studies where there's full data, where we have genomic, transcriptomic and proteomic data sets complete. There were some in TCGA, there were some lung cancer data sets where we have DNA sequencing, we have RNA sequencing, and we have proteomic data. The depth map cell lines. Do you know what the depth map cell lines are? Okay, this is a freely available resource on the depth map. Basically, and the Broad Institute has taken a whole load of standard cell lines, literally hundreds of them, and they have undertaken RNA-seq on those and profiled them, and they have undertaken proteomics on some of them. Now, why is this important? Well, the depth map cell lines are cell models that are frequently used to test if drugs are working or not. So the cancer cell lines, the MCF7 breast cancer cell line is an example. So with these depth map cell lines, because they've been profiled, people are treating them with a drug and then getting an efficacy measure, and then trying to look at how the gene expression or protein expression is influencing efficacy. But more than that, I think it's still part of the Broad Institute, they've done a project where they've taken around 350, 400 depth map cell lines and then exposed them to about 30, 40, maybe even 70 chemotherapeutic agents. So what they've got is they've got the response to chemotherapy, and they've got the expression profile prior to chemotherapy. So that data can be mined in order to understand what features are driving sensitivity and resistance to chemotherapy. So there's the potential to explore that and look for personalized medicine. The problem is the cell lines are not as representative of the tumor as real human tissue. So what we've been doing is modeling efficacy and then taking that efficacy model into TCGA data, which is blocks of tissue, and then predicting across a population what the spread of efficacy might be. So it's extrapolating from a cell line which is probably derived from one person's tumour up to a population of 1,000 individuals. So we can start to explore virtual clinical trials by that method. We can try drugs out by modelling them. So what do we do with this? If we've got a workflow where we have integrated data, how do we use it? Well, first of all, we gather the data. We need to see what sources of data there are out there. We need to look into repositories, GEOD, TCGA, Array Express. Up until this point. If you remember, we can check for batch effects using something like Disney plots. We can normalize and scale data. Well, if we've got FASTQ files, then we can normalize that data and align that data using something like Galaxy. So the SRA, which is the NCBI's RNA-seq or an RNA-seq repository, you can download stuff straight into Galaxy and then run a Galaxy workflow on that, converting the FASTQ, running it through quality checks, trimming it, and then getting counts. I use Callisto, to be honest. The latest method is Callisto. It's a lot quicker than some of the previous methods like string tie and ball gown and top hat, and so it's string tie and mental block. But These methods, Callisto is a more efficient method and a more accurate method. It's been shown and quicker. So the next step then is we're starting to enrich features. So we have data from each one of these pools, and we're addressing the question if we've got the same data across different pools. So we can, if we've got the same pool of data, we can create a meta table, a meta data set, which has copy number variation, methylation, proteomic data, transcriptomic data, even metabolite data. And what we can do is mine that whole meta string, and as long as we know where the feature has come from, which pool, we can then say, right, well, this is the combination of best features that are stable that predict this thing. So we have, effectively, we might have things where we've got methylation elements turning up, we've got transcripts, we've got proteins turning up as all important in the thing that's defining the disease. And we've done that as a group in Leeds University in the UK, where we've taken multiomic data, copy number variation, expression, methylation, and we've effectively pulled features from across that workflow. Another study which we've done, which I'll mention here as well, is there are publicly available data sets for Crohn's, for ulcerative colitis, as a part of a broader group of IBD. In that study, they've got proteomic data, they've got transcriptomic data, and they've got metagenomics and metatranscriptomics data from fecal samples. So they've got the gut microbiome there as well, which means that you can start to understand the interplay between the gut microbiome and the transcriptome of the host. So that's another level of dimensionality, but gut microbiome plays an important role. The point is, if we're doing this integrated analysis, there is no bias introduced. We're considering everything on an even keel. So once we have our enriched features, we can then identify the interplay between these things. So what's the interplay between a gene, methylation, and proteomics? And I said we don't really know how proteins and transcripts link. Well, this allows the potential for us to see how things are interacting that define the expression of a protein. Is it a methylation event? Is it a microRNA event? Are these things coming together? Is there agency from a third or fourth gene that's regulating the production of a protein? So we can start to dig into that. If we wanted to be really detailed, we could take a protein as a target and look across all the multiomic data sets and see which things were influencing just that protein level. And that would start to find associations that might be driving the expression of a protein. So many experiments, only so little time. What we can also do, because we have genomic information, is we can look at mutational states of genes. So we can add another layer of complexity by rerunning this whole process for different mutational states of the gene. So how is this big interplay happening here for the wild type, and how does the mutation of a gene perturb all of that? Is there compensation? Do other proteins come to the fore? Do other genes come to the fore? Is there an attempt by the system to methylate a mutated gene or a set of genes resulting from that mutation? A set of transcripts resulting. How is this working together? This is a very complex system, but we have to be a bit creative and think, how can we interrogate this? So that's the workflow where we have integrated data. Just bear with me, I just want to see if we... Oh yeah, that's good. How do we have what workflow do we create for single cell data? Well, we gather the data, normalized scaling, batch effects. In single cell data, we have to remove dead cells, senescent cells, cell fragments, artifacts, and cell nets with low numbers of expressed genes. So we're kicking out the rubbish. There's huge amounts of debris in any cellular model. If you've ever looked at flow cytometry data, there's so many fragments, and that makes a big chunk of the spectral distribution. So we've got ways of assessing the profile of a single cell and eliminating it if it's got some problem with it. Now, what we then do is we've got all this mixture of cells coming together, and we have to determine what a single entity is. So, the best available technique at the moment is CyberSort. That used to be free, they've just started charging, which is a real pain in the backside. But with CyberSort, you can say what type of cell it is based upon, they kind of use multi-dimensional scaling to derive this, but then histological evaluation of the cells found. But then, effectively, they've got a set of algorithms that look at transcriptomic markers to define what type of cell a given cell is. So it might identify it as an immune cell, it might identify it as a specific T regulatory cell, it might identify it as an epithelial cell, as a mesenchymal cell, as a stem cell. But CyberSort is there to try and identify what type of cell it is. Anybody think there might be a problem with that? What could the problem with that be? Is CyberSort right, do you think? Any ideas? Be brave. There's no right or wrong answer. Is CyberSort right?
What parameters does it differentiate?
Exactly. It's a model. And okay, if everybody uses it, it becomes a very strong model, but it is still a model. Where does it fall down? When does CyberSort get it wrong? And again, this is a key point here. We always think about the workflow you have and whether that workflow is right or wrong. What is the error introduced by using CyberSort? Now, if you use CyberSort to predict cells, in order to look at the profile that defines a cell type, you get a self-fulfilling prophecy, because if you use CyberSort to predict, say, Tregs, and then look for the molecular profile of Tregs, well, those cells have been defined by CyberSort, they will always be Tregs. And the markers you find will always be Treg markers. So you end up with this reinforcement of an idea. So you have to be careful with any method and consider what its limitations are, and clearly communicate the model you've used. In the next 5 years, somebody else might produce something 10 times better than CyberSort. Does that mean that's all results previous to that are wrong? Now, be cautious. There is no right or wrong answer. There are merely ways of looking at a very big world through very tiny windows. That's the problem. Science is like taking a tiny peek at something that's the size of the universe, but through a window of 1 millimeter diameter. So be mindful, be cautious of what is happening in a data set.
Yeah. We can explain how CyberSort actually works. Like what parameters does it take?
It's a classification model. So basically, they've looked at regions of a multi-dimensional scaling or Tisney plot from a mixture of cells, and then they've defined regions and then looked at the genes that are differentially expressed from other regions and said, okay, well, this is a mixed cell population, this is a population of immune cells, what have we got in there that's different from every other cell? And then they'll dig into the immune cells and say, well, what types of immune cell do we have here? But it is, I've oversimplified it, but they've done that extensively across multiple single-cell RNA-seq samples. They've enriched for certain cell types and then run it. So they're not taking every cell. They might, for example, take an immune cell cohort that they've separated out for various means. Then they'll profile that. But ultimately, they're looking for effectively determinants of a cell type and building that into a classifier model. That gets better all the time, but it's still a model, and every model, as I said the other day, has its errors. Is it a paper plane, or is it a full-size model of a Spitfire? That's the question you have to ask. So,
isn't it like in the manually way, like I've done single cell analysis. So what we do is like we do clustering, and from the literature mining, we retrieve all the markers or cell type identifiers which are broadly categorized, like B cell, T cell, or macrophage, and then we extract those specific clusters separately, and then we recluster them to look for the subtypes.
Yeah, that's exactly. You can do that. That's a good way of doing it.
That is like it's a good way instead of using CyberSort.
Well, you can use the literature. CyberSort probably uses literature to some extent as well. But yeah, as long as you've got a rational approach that you can state and be aware of what the limitations of it might be, then that's perfectly valid. Yeah. The challenge is you're looking at something quite complicated and vastly oversimplifying it. And so, for example, with clustering, what form of clustering do you use? Because if you use hierarchical clustering on a whole transcriptome,
basically k-means clustering.
K-means, yeah, that's what I would use, yeah. So if you're using k-means clustering, that's good, because that deals with the high dimensionality. If you're using hierarchical clustering, there's as much distance between the individuals on a branch of a cluster as there are, so you get these these massive differences between individuals, but very small differences between the cluster. So the inter-individual distances are huge, the inter-cluster distance differences are really small. So be careful of using hierarchical clustering on large-scale omics data. So yeah, but that k-means is a good way of doing it. So it is a mixture of clustering, a mixture of evaluation, marker definition, and CyberSort have done that, but you can do that manually. If CyberSort is a quicker way, because as I'm sure you're aware, going through the whole clustering business and defining the markers for types is quite laborious. Yeah. But it is an accepted method, CyberSort. So how can we use single cell work? Well, if we use that concept of a digital twin, you remember I showed that yesterday, and we're quantifying a pathway, what we can do is say, right, let's build a model of this pathway for mesenchymal cells, for epithelial cells, for stromal cells, for cancer cells, for stem cells, and how do those models differ? So, as an example, there's a piece of work I did for a large pharma company, and they were interested in autoimmune disease. We were right in the final stages of writing this up as a paper with them, so I'm not revealing any confidences. So they were interested in this autoimmune condition. They'd got lots of blood data, they'd got lots of tissue data. So they had a hypothesis that there was a particular pathway, or two particular pathways, that were of interest. So they wanted to validate that hypothesis. So what we did was, the first thing we did was look at disease versus healthy for blood and tissue. So we ended up having an enriched set of markers for blood, for the disease, and an enriched set of markers for disease versus healthy. What we then did is use the pathway method that I showed you yesterday for both of those pathways, and enriched around the pathway for disease, blood, and disease tissue. So this gives us a list of features for phenotype of disease and a list of features for the pathway. So in one pathway we found we had 86% overlap with phenotype, so that's good. That pathway is valid. In another pathway we had 6% overlap. So they were surprised by this because they thought both pathways were relevant. So we'd validated that one pathway was there in bulk tissue and bulk blood. Where things got interesting is we then repeated that analysis for single cell of tissue, and we were looking at we looked at fibroblasts, we looked at infiltrating lymphocytes and epithelial cells. We then enriched the phenotype and enriched the pathway, and what we found was the pathway, which only had 6% coverage in bulk tissue, when we looked at infiltrating lymphocytes, we had 90% coverage. So the thing that was missing in bulk, when we went to specific tissue types, became apparent. So you can see how we can reveal mechanisms and validate mechanisms by looking at different types of data and seeing which context works and which one doesn't. So we then validated that pathway, but it only validates for one cell type. It doesn't validate for all the other cell types. So what was happening in bulk is that signal was being diluted. But when we look at single tumor infiltrating lymphocytes, it was strong and it was there. So that's an important thing to consider. We can look at single cell data versus bulk data and cross-compare the two. So we can run network inference on the different cell types, and we can, if we have a pool of cell types, let's say we have stromal, we have epithelial, we have tumor, we have infiltrating lymphocytes. If we take the signal, let's say we've got transcripts, and we label the transcript name with where the cell came from, and then we pool that into one big data set, we can start to enrich around that and see, do markers from different tissue types in this mixture of tissue talk to one another? What is the strength of interaction between a T cell expression of IL-10 and a stromal expression of EGFR? That's a simple example. But by labeling with position, we can start to look at tissue crosstalk and to see whether there is crosstalk between cell types. That's of importance. So that's another layer that we can build into this if we want to. Types, and how is it differential between a mutant and a wild type state of a gene? So, you can start to see, this gets complicated, there's layers to it, but we can try and disentangle those layers. Any questions? Hopefully you can get a flavour for how all this multiomics stuff that fits together, how we can use it to probe and interrogate data and understand what's going on. Is it clear for you so far? Are you okay? Yes? No? Okay. The next thing we can consider is spatial omics. So nanostring. So spatial omics is also being, does anybody know about 10x single cell sequencing? So 10x have a new method called Infinium, and 10x Infinium is looking at single cell, but it also tracks position in the tissue and is looking at spatial single cell. Just another layer. It goes beyond what nanostring does. But we can do the same sort of thing. But here we're looking at regions of interest. And then basically we label the gene name with region of interest. So can we look at the crosstalk between a set of tumor cells or between a tumour region and a stroma region. Much in the same way as single cell data, by labelling with region of interest, we can start to explore the interaction between these things. If we don't have integrated data, we have a bit more of a problem. Here, what we have to do is we have to enrich for the data sets separately. So we would enrich separately for transcriptomic, proteomic, methylation, metabolomic. So that gives us a pool of things for each one of those compartments, probably from a different population, because the same experiment hasn't been done on the same cases. But what we can do here is start to look for the co-occurrence of things. So does something crop up in the gene at the transcript level, and also crop up in the methylation level, and also crop up in the proteomic level? But we can't really say these are competing with one another, because the answer is coming from a different data set. So it's harder to do integration unless we've got a contiguous data set. So contiguous is where we have it all joined up. It's the same thing for, same set of cases for genomic, transcriptomic, proteomic, methylation, metabolomic. But we can look for commonalities and run the network inference on the commonalities. So, one case study, we did this some time ago. This is I think it's a European Journal of Cancer paper. We did this 2012 actually. And what we were doing is looking at array data, and for the same cases they'd got microRNA arrays and cDNA arrays. So they had integrated data, they had mirs, they had transcripts. Of course, now with deep sequencing, we have the mirs, we have the transcripts anyway. So they're all automatically there, so we don't have to worry about integration. So here we were looking at nodal status and estrogen receptor status in breast cancer. And we'd enriched for mirs and cDNAs. And then we ran network inference. And what we found was that certain mirs, we could see how the mirs were influencing the expression of genes, and how that was different between ER positive, ER negative, luminal A, non-luminal A. So we could work out what the drivers of that system were, but how those drivers were being modulated by microRNAs. And that's important because they're playing a key role in the whole system. So, second case study, and this goes in a bit more depth, is what is the impact of the TP53 mutation on the TP53 pathway. So here we got breast cancer data. This is unpublished. We got TPM values, and for the breast cancer data we knew whether they were wild type, missense or nonsense. We knew the specific mutational points, but so we then translated to those where they were known commonly as missense or nonsense. So we used the pathway miner bit, where we look at the addition to the pathway, to kind of explore what was impacting on that pathway. So, but you know how I showed you we could mine a pathway? Well, we were mining, in this case, every single gene in the P53 pathway and looking at what interacted with it stably. The key thing is what interacted with a gene of the P53 pathway stably, because some things, if they're not stable, they won't be interacting. Now there's a lot of data here. I'm only going to show the tip of the iceberg. But what we were able to do with this is, because we had a map of what was interacting for every gene of the P53 pathway, we could see firstly how many stable features there were interacting with the gene. We could see what interacted with the gene, and we could see whether the gene, for an unseen cohort, predicted itself in the top 500. That's important because a gene, if it's stable across a population. You build a model to see how it predicts itself on training data, but that same function should carry on into unseen test data. So by using the performance on an unseen data set, we could see how stable it was across the population. And in some cases, as you'll see, the gene that was the output of the neural network model didn't even predict itself in the unseen cohort. What that means, it's highly unstable. There's message coming out of that gene, but it's complete nonsense. So we're able to effectively test the stability of a gene's interactors, and we can also test that stability in a healthy state as well. I'll illustrate what I mean. So this is the gene list. Here, instead of using the KEGG data set, we use the hallmark. There's a few more genes in the hallmark list. So what did we see? Well, in the wild type, there was a strong signal coming from P53. We saw 402 out of 500 things interact. So we're looking at the top 500, we're sorting based on the unseen performance, and then we're repeating that in 20 different cross-validations. And then each one we look at the top 500. So if something comes up, if it came up 500 out of 500 times, that would mean across the 20 repeats we'd have perfect consistency. Everything is coming up stably. If something comes up less than 500 times, then there's minor instabilities. Here we had things coming up 402 out of 500 times, which is pretty stable. The things that were coming up were pretty consistent and stable. Does, in this case, P53 predict itself across the multiple data sets? So remember, in that list of stable interactors, ideally we should see P53. It should be predicting itself. And it did. And then if we looked across the whole pathway in the wild type form, how many additional new features were there? Well, in this case, if we look at all that stability, there were 2,652 new stable features coming into that broader pathway environment. So again, that suggests that it's mapping a very comprehensive and strong signal. So that was what happened for wild type. If we looked at missense, so this is a disruption of the signal. It's noisy. In missense, how many P53 interactors did we have? 1. So we had 1 interactor out of 500, and that wasn't P53. So we found something which is probably a false discovery. P53 does not predict itself. So in the missense setting, P53 was highly chaotic. It's producing loads and loads of signal, but the signal's complete rubbish. So P53, in this case, is not doing its job. It's not predicting itself. It's losing information. If we look at the number of genes in the stable features that we found, we only found 90. Those 90 are probably compensatory mechanisms dealing with the loss of any signal or dealing with the aberrant signal coming from P53. So that's the mutated missense state. We also had a non-sense state. Here is kind of a halfway house. We saw 245 things predicting P53, so it is something's happening here. It might just be that it's a low level of expression. It's not completely deleted. It's effectively not much signal coming from it. P53 did predict itself, and we had 393 genes stable interactors. So we've got a weak signal here. It's not as clear as the wild type. It's less stable, but it's less stable because there's less signal. So with this, we were able to effectively say we ran all these lists through network inference, and we were able to say what perturbations arose in the pathway using a hypothesis-free, evidential-based approach, and how different mutational states perturbed that. Now that's important because if you want to find a treatment, if you know you've got a patient who's wild type P53, then you can find treatments for them that will help deal with their cancer in the context of wild type P53. If you've got a missense P53, that creates a different state, and you're dealing with the compensation system where P53 signal has been perturbed, messed up. So you treat that patient in a different way. This suggests where you could treat them. And then the nonsense setting, again, you have a different setting where you would treat those differently. Now there was a small degree of overlap between these three. There were common features, but there were specific features to each one of these states as well. So we have the 2652, but there were not many of those 90 that overlapped. And there were a few of the 393 that overlapped as well. So we were able to see where the overlaps were, where the commonalities were, and where the differences were. So that's ending this lecture. Any questions? I've kind of taken you to the tip of Everest in terms of the amount of stuff we can do with omics data, so it's probably a lot to take in, but what I would say is read the notes, contemplate it, and see where you get to. But any questions now? No? Okay, what's the time? Right, I think we can probably go to the last lecture now, which is not a huge one, and that will mean we've done my lectures before lunchtime, and then we can start getting to other things in the afternoon. Right.